And good afternoon. My name is Klaus Pettersen. I'm a physicist who turned into a theoretical neuroscientist, turned into a leader of the Norwegian National Artificial Intelligence Research Consortium, NORA. But today, I have the honor of chairing the Brain Prize lecture. So the Brain Prize is awarded each year by the Lundbeck Foundation. The Lundbeck Foundation is one of Denmark's largest foundations, encompassing a comprehensive range of commercial and philanthropic activities, all united by its strong purpose, bringing discoveries to lives. The foundation's philanthropic grants amounts to more than 500 million Danish kroner annually and primarily focus on the brain, including the world's largest personal prize for neuroscience, the Brain Prize. The Brain Prize recognizes highly original and influential advances in the area of brain research, from basic neuroscience to applied clinical research. The Brain Prize 2024 was awarded to Larry Abbott, Terry Sejnowski, and Chaim Sompolinsky. All three are originally physicists, and I guess all three still consider themselves physicists. However, they are obviously much, much more. Professor Richard Morris, chair of the Brain Prize Selection Committee, explains the reasoning behind this year's award. Theoretical and computational neuroscience permeates neuroscience today and is of increasingly growing importance. Larry Abbott, Chaim Sompolinsky, and Terence Sejnowski have made pioneering contribution to the field and have made seminal discoveries in our understanding of the principles that govern the brain's structure, dynamics, and the emergence of cognition and behavior. All three candidates originally trained in physics, but they have long worked in neuroscience and have applied novel and sophisticated approaches from physics, mathematics, and statistics to the analysis of highly complex datasets acquired by experimental neuroscientists. They have also proposed conceptual frameworks for understanding some of the brain's most fundamental processes and how these may go awry in some of the most devastating disorders of the nervous system. Their scientific achievements have also paved the way for the development of brain-inspired artificial intelligence, one of the emerging and transformational technologies of our time. In his own words, Terry Sejnowski is known as a person who builds things that actually work. I saw this in an interview, Terry. A puzzle solver who was fascinated by the heavens, the stars, and the galaxies. He started as a master student with the legendary physicist John Wheeler at Princeton University as a supervisor. He then went on with a PhD with John Hopfield as a supervisor, graduating in 1978. I should say that although he was a trained physicist, his first paper in neural networks was already in 1976, the year I was born. And I am not young anymore. He did theoretical or computational neuroscience long before this was even a field. He was a postdoctoral fellow at Princeton University and Harvard Medical School before being appointed to a faculty position in the Department of Biophysics at Johns Hopkins University in 1981. He moved to La Jolla in 1989 and is currently the Francis Crick Professor at the Salk Institute for Biological Studies and Distinguished Professor of Neurobiology at the University of California, San Diego. Terry Sejnowski was also an investigator with the Howard Hughes Medical Institute from 1991 to 2017. Professor Sejnowski's research aims to understand the computational resources of brains and build linking principles from brains to behavior using computational models. He pursued his goal with both theoretical and experimental approaches at multiple levels of investigation, ranging from biophysical to systems level. His laboratory developed independent component analysis for blind source separation, which is universally used for analyzing EEG from the scalp and brain imaging by functional magnetic imaging. I should also mention that ICA method is most famous maybe for being the first practical solution to the cocktail party problem. And this can be useful later today, where you separate the different kind of parallel conversations. Professor Sejnowski was also a pioneer in developing learning algorithms for neural networks in the 1980s, inventing the Boltzmann machine with Geoffrey Hinton. This was the first learning algorithm for multi-layer neural networks and laid the foundation for deep learning. NeurIPS organizes the world's largest AI conference, and Sejnowski has been part of the organization since its start in 1987, organizing the 1988 event and being a president of NeurIPS since 1993. Sejnowski is also leader of the recent convergence between neuroscience and AI, the field often nowadays referred to as neuro-AI. Sejnowski said in an interview that the prize is of course important for him, but more important for the field, and especially for the younger generation in the field. It gives the field of theoretical neuroscience an importance, and also the young generation. Terry Sejnowski has inspired so many to go into theoretical neuroscience and so many to go into AI. I know this very well because I am one of them. So with that, I'm really looking forward to your talk, Terry, titled Brains and AI.
Well, thank you very much for that introduction. With the time remaining, I have some, I'm really pleased to be here. By the way, last night at dinner, I was reminded by Roger Nickel that my first visit here was over 20 years ago to a workshop organized by Per Anderson. And he took us to his cabin off in the hills. And I remember that because it was a small group and we'd had these great discussions. And I've visited many times since then, up in Trondheim to visit the Mosers. But most recently, I visited to visit Lofoten, not for a scientific meeting, but to climb the mountains there. And so saw a different part of Norway that I really loved. And so I'm really pleased to be asked to come back here. Now, we're living through an extraordinary moment in history. Two revolutions are taking place at the same time. As you probably know, there's been a revolution in AI in terms of the models that are being used today, which are quite different from what was at the early stages in the 20th century, based on symbols, on rules and logic. Models today are based on very loosely in terms of simplified cortical architectures, deep learning. And there's another revolution that's occurring in neuroscience, in systems neuroscience. In the 20th century, neuroscientists recorded from one neuron at a time. Now, it's painfully slow, but also it gives you a very myopic view of how the brain is organized on a larger scale. But now we can record routinely from tens of thousands of neurons, from multiple parts of the brain, dozens of areas simultaneously. And that has given us a global picture, which is a very different one from the conceptual framework in the 20th century. And that's what this talk's going to be about. But even more, I think, important is that now that we have a better model for AI, okay, I'll take that as a compliment. These two fields are converging. There's some overlap because we can talk about processing units and neurons. They're not the same, but the architectures are similar. And you'll see in this lecture that we can go back and forth in terms of inspiration. Okay, so by the way, this is what it looks like outside my lab at the Salk Institute, the Pacific Ocean. Okay, so I want to introduce, first of all, two different modes that brains can be studied. The first one, I'm going to illustrate that by an animation of what may be happening in a monkey brain when the monkey is hungry. But the idea, though, is that when neuroscientists study a monkey, they study it in the lab and typically give a stimulus and record from neurons throughout the cortex and look for neurons that are correlated with, say, the sensory input or decisions or the motor output, right? And that whole process takes less than a second. And you'll see some of the markings of the times it takes are listed here. But I want to show you what happens before the monkey has encounter with a prey. Monkey's hungry, right? It's in the jungle. It has to figure out where to go. Where can I get something to eat? Well, it relies on previous history, experience. And it thinks. It has to think about where's the best place to go right now. Maybe it's the water hole, maybe somewhere else. Okay, so that may be quite a long time. Suddenly, it sees something it likes, and within 100 milliseconds, the visual input has been recognized, and then in another 50 milliseconds or so, 60 milliseconds, the motor system is activated, the muscles are contracted, and the prey is caught. Okay, now then what happens? Okay, the monkey has to decide, where should I go? Should I share the food? In other words, it's thinking. Now, it's the middle sequence there that neuroscientists have studied, and we understand a lot of what goes on in the different parts of the brain. But what happens at the beginning, most of the time, monkeys are not responding to the stimulus. Most of the time, they're making decisions, socializing with other monkeys, thinking about, where should I go? What's happening? Is there danger? I mean, these are all things that are not being immediately stimulated by the environment, but by their past history, the way that we do, very freely moving. So here's the question now that I'm going to try to address here is, how is it that brains do that, this processing that's going on in the background, thinking? How do they integrate, remember, plan, and act on sensory inputs over hours? It's a time scale that's very difficult to study. If it's a second, you can record and look at the responses of the neurons. But what do you do when someone is sitting in a room and thinking, what should I do tomorrow? What happened yesterday? Right? There's no overt actions taking place. There's no sensory input. So how do you link what's happening in the brain to all of that cognitive processing? That's it can be done, but it's much more difficult to do interpretable experiments. And what I want to do is understand the neural mechanisms underlying that process. And here's an example of something that's going on in your brain right now. Okay, you're going to hear my lecture. You're hearing word after word after word, slide after slide after slide. You're taking all that information in, and you're interpreting it in terms of your previous knowledge base, your long-term memory. And then at the end of the lecture, you'll think about it, and maybe you'll ask a question. Where is that happening in the brain? How is it happening? We don't know. We don't know the neural mechanisms. We can speculate, and there are some really good evidence I'm going to show you, but it's still an open question. Same thing happens when you read a book or an article, right? You can read it, and at the end, you can summarize it. You can say, well, here's the story I just read, right? How did you take that in? By saccadic eye movement, 3 times a second. Bang, 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 bang. You're integrating all that information, all those words, one word after the next, a temporal sequence. But somehow you're able to now see the whole global picture of what was described in the article in the book you just read. If you think about it, it's the same problem. One is auditory and the other one is visual. Now, the psychologists, cognitive psychologists, have studied this, and here's the way that they summarize what I just described behaviorally. Well, there's short-term mechanisms for taking sensory information in, and short-term mechanisms for sequences of actions. But the part that is the most mysterious here is the long-term working memory. And this is what's taking in all that information. You can rehearse it to remember it. It's transient. It only lasts for a few hours. And the next day, you may not remember what you heard in my lecture. You may remember a few salient points, things you attended. Or maybe you'll remember the question you asked. But you won't remember all the slides. Now, the working memory is really critical. It really involved with the long-term memory. You can retrieve things selectively and put that into your working memory. So that's what we're going to try to understand. If you're interested, I wrote an article here. The transmitter is a website sponsored by the Simons Foundation, and the essays for the general public on neuroscience, and I wrote one called Thinking on Thinking, which sort of summarizes this lecture, but in a very general way. If you're interested, you can Google it. Okay, now here's where we are. Memory mechanisms exist from seconds up to years, and we know most about short-term sensory and long-term memory. We have mechanisms for them. Here's the big hole. Short-term working memory is like remembering a telephone number. You can rehearse it, you can remember it for a minute or two. But the long-term working memory, which is based on things you already know, fitting new things into the old structures, that can last for hours. So here we go from seconds, minutes to hours, and this middle section here is the part that we need to fill in. Let's get a hint from large language models. Now, if you, we'll go through this in a minute, but if you've had a dialogue with a large language model like ChatGPT, you start typing in your query, and you start getting a sequence of words in return, and you can go back and forth. And it looks as if the large language model remembered everything that was said, that you said, and what it said. So somehow it has the equivalent of a long-term working memory. Well, let's figure out how did it do that? Well, first of all, here's the query coming in. This is a module. There may be 100 of these stacked up, and it's a feed-forward network, and it's trained to predict the next word in a sentence on trillions of words, huge database. Now, what happens is it responds to your query with a word, which then loops back to the input, and then it goes around and around, one word after the next, after the next, after the next. Okay, is that familiar? Yeah. It is familiar. It's what happens when we have a dialogue with another human being or reading a book. Now, the way it solves that problem is taking the temporal sequence and converting it into a long input vector. Every word is added to it, so it gets longer and longer and longer. So it actually converts a temporal sequence into a long spatial vector. Okay, well, that's something. Can we do something equivalent to that? Well, we don't have a perfect buffer of everything that you've heard recently, but is there some way that the brain can do something similar? Well, we have something similar with regard to this loop. Here's the cortex. And the models, the deep learning models, this feed-forward network here is the model for the cortex. But we know in the cortex that there is a loop projecting down to the basal ganglia topographically and then back through the thalamus to the cortex. There's a loop here of 100 milliseconds. And have you ever asked yourself, if you're talking to somebody, where's the next word coming from? It's coming from your basal ganglia. It's automatized. And it is in order to be able to use it, it has to learn sequences like playing tennis. You have to practice, practice, practice. Well, talking requires a lot of practice, right? You whole developmental period during which you're learning language is practicing and training your basal ganglia so that it can use language in a very natural way. And it has the same loop structure, okay? Well, that's interesting. I'm going to come back to this at the end of the lecture. Well, let's look for mechanisms. Where in the brain are mechanisms that could convert temporal sequences into spatial ones? Well, we know this is a log time scale here, milliseconds, seconds, hours. And we know a lot about the fast things, the electrical signals at synapses, which is, where we think a lot of the memory is stored. But we also know something about the long-term mechanisms, like long-term potentiation, long-term depression, that presumably last for, our lifetime. But what about this whole area here between seconds and hours? Well, it's a black hole in the sense that we don't know of mechanisms. No mechanisms have been identified, but there has to be. Otherwise, we wouldn't be able to think, right? And so we want to fill that gap. Well, here's a mechanism. We've known about oscillations since Hans Berger recorded the first electrical signals from the scalp. This is the alpha rhythm, and related it to behavior, whether you're relaxing or where you're actively working with the sensory motor system. Now, here's there are rhythms all the way from 2 hertz up to 80 hertz, different frequency bands. The gamma oscillation, as you can see here, recorded with field potential and unit activity, lasts about 200 milliseconds. It's a burst. When you pay attention to an object, you get a burst of gamma oscillations like this. You can see here, this is the frequency band. And the oscillation suggests that it's synchronous. But if you can record it from one electrode, the question is what happens if you have an array of electrodes? So here's 3 electrodes that were spaced by 0.5 millimeter during one of these gamma bursts. You can see the peak moves across the cortex. It's not a synchronous oscillation, it's a traveling wave. And it turns out that every oscillation is a traveling wave. Now that we have arrays of electrodes we can record optically, they're all traveling waves. And it's not just, occasional. You get traveling waves that can be stimulated by sensory input. You get spontaneous traveling waves. Attention can activate it. And so this is interesting because, traveling waves are spatial as well as temporal. Well, space, right? Well, that's something that we might be able to use for long-term working memory. So let's think about a stimulus, a visual stimulus, and say in V1, primary visual cortex. Well, we know that that produces a traveling wave that is shown in this ring over the 40 to 100 milliseconds, depending on what part of the cortex you're looking at. But in the early parts of the visual cortex, it's measured in tens of milliseconds. Now, you might think that, gee, you've taken that stimulus and now it's spreading out. You've lost information about where it started and when it started. No, you haven't. Because if you look at this ring, by looking at the size of the ring and knowing the velocity, you know where it started. The information is still there in the activity of that ring. And the position is the center. Right? So you haven't lost the information, it's still there. It's been recoded into a different pattern of activity that mixes time and space. And we call this a space-time code. And just to make it explicit, this is a TINS paper that came out last year. Here's an input coming from the thalamus into a cortical neuron. And it is now being mixed with information that came in an earlier time into another neighboring neuron that through long-range horizontal connections, which are unmyelinated and are very slow, then impinge on A and mix with information occurring right now. And the conceptual framework is that if you look at V1 and you get a snapshot, that's the information of what's happening right now. But because of the fact that we know about all these horizontal transforms and traveling waves, that it's not telling you about what's happening right now. It's telling you about what's happened in a time window. It's mixed space and time in a very interesting way. And as you go up the hierarchy from V1 to V2 up to infratemporal cortex, say, the receptive fields get longer, larger, and also the temporal window gets longer. And in fMRI experiments that were done by Uri Hassan, by giving brief clips, he showed that the timescales in V1 were on the order of a few seconds, and by the time you get up to the prefrontal cortex, it's on the order of a half a minute. So this is literally recoding information so that you now can work with information that has happened in the recent past. And 30 seconds is actually a really long time period if you think about it. So I mean that's a different way of recoding the information that's similar to what's happening in a large language model. Okay, let's make a model. The cortical connectivity has a Mexican hat structure like this, where local connections are excitatory and more distant ones, neighboring columns, is suppressive or inhibitory. And if you make a little input-output matrix where the colors represent the strengths of the synapses connecting the pre and the post, it looks like this banded structure, and that cross-section is this white line. But you'll notice that along each diagonal, the value is going to be the same. It's excitatory, peaks here, and I'm going to come back to this and just notice that it's diagonal, right? And this ring model, where I'm going to show you about, is just basically taking a bunch of units, connecting them up onto a ring, and seeing what the dynamics looks like. And it turns out that this model was used earlier by Ketchen Zhang, who was a postdoc in my lab, to develop a model for compass cells. In the hippocampus, the rat knows what direction it's pointing, the head is pointing. And there's a bump of activity, basically, that moves back and forth along that line. That line is actually the compass in the case of the rat. Very well studied. Well, we developed a ring model for the cortex that takes into account not just the neighboring connectivity that you saw in the last slide, but also the time delays, those long-range horizontal connections. And when we put them together, we were trying to get these traveling waves to come out. And traditional models in computational neuroscience, typically have a few hundred, maybe a few thousand units. We could get very nice dense traveling waves where all of the units were active at the same time as the wave went through, which is what happens with epilepsy. But it turns out with the traveling waves in the cortex, with these, in these frequency bands, it's sparse. Only a few percent are active as the wave goes by. And it wasn't until we got up to 100,000 units that we started getting something that looked similar to what you see in the cortex. And I'm going to play now a movie. It's a spiking network model with that connectivity, and what's shown here is the excitatory inputs, the membrane potential, so to speak, for that, that network, and as it dynamically moves in time. Okay, so you can see these traveling waves moving. Now it's bidirectional, this is symmetric. And it really has a very, very wavy structure. And you see things going back and forth. And here's something where they all go down at the same time, but then you start getting waves coming up, going in both directions. And so this is, when we saw this, we realized that, you have to do simulations on very, very large models to come close to the connectivity pattern and dynamics of real neurons. You can't use small networks to understand this. Now, this is what a two-dimensional model might look like. Here's a patch of the cortex, a two-dimensional, connected locally in all the directions according to this activity, connectivity. And you can see just a few in blue dots here. Those are the ones that are active as the traveling wave goes through. That's what we're trying to understand. Okay, we can model this. This is now a machine learning project that was done in collaboration with Andy Keller, who was at the time a graduate student with Max Welling in machine learning. And this is a machine learning conference that took place earlier this year. Okay, so the question is, can you train a network to remember sequences? And the answer is yes. It's been done for a long time with something called recurrent neural network. And just to tell you, roughly, this is a very simple model where there is the next time step, the activity in the network is updated with this nonlinearity, a sigmoid nonlinearity, by a matrix then that acts on the previous time step and then an input x. And if you put in these are the connections to get a traveling wave in one direction. You can see one is basically above the diagonal, and that's what representing this differential equation that is discretized. Okay, so, and by the way, recurrent neural networks had been trained, for example, to do language translation, and they're always started with small random weights. And you train them with backprop through time, which is not biological, but it works. And at the end, you get recurrent neural networks that can do amazing things. So we wanted to compare, starting out with this, compared to the traditional way, which is small random numbers, fully connected. And here is what you get if you take the fully connected version. You can see local correlations, but there's no global organization. But here, with the convolutional recurrent network that I just showed you, you get beautiful traveling waves that are coherent across the whole recurrent network. Okay, well, let's do a task just to show you, what you can do with it. This is called a copy task. You're given a sequence of inputs, and I think in this case there were like 100 units. And then after a certain time, here the delay is 0, you train the network to reproduce the input, and it does it perfectly. Let's put in a time delay of 10. Here's a new input pattern. It reproduces it perfectly. What about 80 or 30? Here's the pattern. Here's the reproduction. Okay, it does it perfectly. How about starting with small random numbers? Okay, this is that network. Okay, 0 time delay. It's struggling. By the time it gets to 10 steps, it's lost it completely. It doesn't hold on to the sequence. It cannot preserve the information. Now, not only is the performance much better, but look at the error as a function of time, training error, mean square error. Okay, so here in red is this convolutional network with traveling waves, and then in these colors are 2 different networks that do different sizes with random initialization. This is log scale. It goes literally over from 10 to the minus 1 to 10 to the minus 7 within the first 100 steps. It's learning orders of magnitude, 2 or 3 orders of magnitude faster. And it does that, as you can see, for 30 and for 80. And here, the network doesn't even get off the ground because the delay is much too long. So this is amazing. So it means that recurrent networks with the right initialization of traveling waves can do not only copy the task, but also all kinds of interesting problems that have to do with keeping track of input numbers, language translation. A lot of different tasks can be done orders of magnitude more efficiently. Now, there is a big oscillation in the hippocampus, as you all know, the theta oscillation, which in rats is on the order of 4 to 8 hertz. And we know that the hippocampus is in the business of learning sequences. We know that from recordings. Not only does it remember the sequence of place fields in the environment of the rat, it also can replay it at night. Right? So this is a wonderful place to look. And we have a neuron paper where we showed you can do that starting with small random numbers. It took a lot of training, but we got it to work. And it's I don't have time here to go into the paper, but just to say that Sejnowski, this is now talking about 15 years ago, published a paper in which he put an array of electrodes in the hippocampus. And this is what he saw. Color-coded, you can see. There's from dorsal to ventral. You get a beautiful traveling wave across the whole campus. And Yuri Bujaki confirmed this, and it's a half wavelength across the hippocampus, but it's always moving. Wow, hippocampus, it's in the business of remembering sequences, and traveling waves may help it do that, right? So this is something now that we can study. Okay, well, electrical activity, circulating electrical activity takes you so far. Maybe, if you're lucky, seconds to maybe a minute, right? But what about an hour? How are you going to go from seconds to hours? Well, there's a candidate here, STDP, spike time dependent plasticity. This is the discovery paper in Science, 1997, by Henry Markham and Bert Stockman's lab. It was a slice recording from two different pyramidal cells that were connected directly. And you all know the story that if you have a burst of spikes, first on the presynaptic and the postsynaptic within 10 milliseconds, you get elevated long-term potential, LTP it's called, okay, but we'll come back to that. And if you reverse that, so you get the post, then the pre, and pair that, a burst of say 5, repeated 10 times with 4 seconds in between, you get something that reduces the strength of the synapse. Now, okay, so there's a couple of interesting things here. If you read the paper closely, discover something very interesting. Is that, first of all, if you don't repair it 10 times, 10 bursts, and only do it once or twice, it only lasts for 20 minutes. Well, that's interesting. Because, long-term working memory, you don't want to remember it permanently. You want to erase it so that you can make room for something else that you want to think about later, right? So, in fact, it's better not to have long-term. And so STDP actually could be what we're looking for, temporary memory. Number two, you can see that the frequencies of the pairing here, if you're below 10 hertz, nothing happens. This is like 2, and I think this was 4 hertz. But as soon as you get a 10 or above, you can get increases that get higher with the highest frequencies. Here's gamma. That is exactly the same frequency range as traveling waves. Another mystery that I've always wondered is how is it you can get 10 millisecond timing? I mean, rate code is not going to help you with that, right? But a traveling wave is a wavefront that goes across and is precisely timed. And we know that that's actually very important early in development in the retina. Traveling waves across the retina are very important for getting the whole cortical hierarchy, in the thalamus and the V1, getting it all topographically organized. We know that. So why not at these frequencies in the cortex? Now, this is an aside. Donald Hebb published this book, famous book in 1949. I was asked to review it by Neuron in 1999. And the most famous part of the book is this postulate, Carl Schatz has summarized it: neurons that fire together wire together. Coincidence, right? And there's a lot of evidence for Hebbian plasticity. And modelers have incorporated into models, and you can get good associations. I always thought that this was a model, this hypothesis, this postulate, was a model for things like conditioning. It turns out Hebb hated behaviorism, which is, that was the top area of neuropsychology in that era. But Hebb was trying to solve a different problem. He wanted to see how is it that sensory information come in can be maintained. And in fact, here's now a quote from my review. Although vibratory activity lasting for up to half a second had been observed by Laurent Deneau, it went further and suggested that such activity in one cortical circuit through converging projections activate areas of the cortex and lead to a sequence of activity called a phase sequence. So this is actually from Hebb, okay? So this is interesting. By the way, in 49, this is before Hodgkin-Huxley, is before Katz. And this diagram here is one of two figures in the whole book. And it shows the V4 connection from V1 to V2, 17, 18 in the cat, but it also has the feedback too. And this is amazing because the anatomy at the time was not very well worked out. But let's hear what he actually said. He didn't say that it was coincidence. He said when A is near enough to excite cell B repeatedly or persistently takes part, the only way that could happen is if it comes before the postsynaptic spike, right? So it's actually what he has described here as really STDP. Now, he didn't say anything about how you decrease the strength of the synapse, but we now know that there's a way of balancing it, right? If the relative timing, if the order changes. Now, okay, so we have a hypothesis here. We have traveling waves creating a space-time code, and that getting integrated into the synapses and accumulating information over a few hours. Now, the question is, which synapses? There's a pretty good candidate. If you look at the distribution of synapses, it has a very interesting shape here, which has a big accumulation here at very small sizes. And by the way, the spine head is proportional to the strength of the synapse, and that's what's being used as a surrogate here. Now, if you transform that into a log scale, you can see it actually is Gaussian, so it's log normal. Now, here's what we know. We know that the big ones here are the most stable. That's probably the ones for long-term memory. But we know that the smaller ones, that's over 90% of them, are labile. It's very easy to change their strength because they're small, and the calcium comes in and the concentration goes up very quickly, as opposed to these big synapses where you have to have a lot of calcium coming in. And furthermore, some of these small synapses are coming and going. There's turnover. So it could be that long-term working memory has access to 90% of all the synapses in the cortex, excitatory synapses. That's an enormous number of synapses, right? So it has a huge capacity. But it's only temporary, right? It fills it up and it maintains it, rehearses, and so forth. But eventually that will all fade away. And that's what you need if you want to be able to reuse it, your cognitive processing for another task. So I'm going to fill this black hole with a combination of dynamical traveling waves, excitatory connections, excitatory activity, which is coupled with STDP to extend it to hours. That's the hypothesis. Now, what can you do with that? Well, we don't know. And I have a bunch of experimental ways of testing this kind of, it's a really conceptual framework at this point, right? The pieces we have are there in the literature. I'm putting them together to help us perhaps solve a difficult problem. Now, if you go to the literature, it turns out that there is some interesting papers. This is a summary of them in Trends and TINS. And when you do an experiment in which you have a visual stimulus at threshold, so we only see it half the time, reporting that you saw it, what you find is if you take the trials where you saw it, you get activity in the visual cortex and the occipital lobe that spreads to the prefrontal cortex. But if you didn't see it, not perceived, it doesn't spread as far. Well, that suggests that maybe it's the spread of activity globally throughout the cortex that helps you integrate information that's coming in, not just that you've seen it, but that you're actually going to operate on it. And now this led Bernie Bars to this theory he called the global workspace. So the idea is that not only does the information go feed forward, but it also goes feedback. And so now you get global activity that's circulating within the cortex. The feedback connections are very difficult to study, and we don't really understand their function. We think they have something to do with attention, but there's more feedback connections than are feed forward. Okay, so under what circumstances do we have really global traveling waves? Gamma is very local. It's usually typically localized to V1, the borders. And each of the areas has its own gamma. But here's an example of a traveling wave, which is truly global, and it happens when you fall asleep. So in stage 2 sleep, non-REM sleep, you have these bursts of they're called sleep spindles, but they're bursts of 10 to 40 hertz. And literally, this is happening because of the fact that you have ion channels that are activated when the cell is hyperpolarized, which is what happens when you go to sleep. It originates in the thalamus, projects to the cortex, and then creates a global signal across the whole cortex. Now, it was thought, and I collaborated with Mircea Steriad for many years, 15 years, that it was synchronous across the whole cortex. And it's true, the whole cortex is spindling. But the question is, what's the relative timing? What's the phase between the locations? We had an opportunity to analyze data from humans. These are patients who have epilepsy and are drug resistant, and the last chance they have is to open, literally, a bone flap and then to insert an array of electrodes. Here it's 10 by 10. And then you wait for 2 weeks because you need to have seizures to figure out where they originate. And once you do that, you go in and you take it out as long as it's not an area that is important for language or motor systems. And in many cases, it can alleviate the seizures, which are really, really debilitating. You can't drive, you can't do a lot of things because you have no idea when the next seizure is coming. Okay, so here is what the pattern looks like, just like you saw for the hippocampus, but now for the entire hemisphere. And it's normalized between +1 and -1, so just follow the white dots. Temporal, parietal, prefrontal. TPF, TPF. And it does that 10, 20 times, and that occurs thousands of times during the night. And we know that with drugs, you can show that if you increase the density of spindles, you will increase what you remember the next morning if you're trained just before you go to sleep. So it's very likely to be causally related to the memory consolidation that we think happens during sleep. And by the way, the spindles are triggered by the hippocampus. It all fits together. In terms of now setting up global organization. And what could be happening is there are long-range association fibers which can have time delays, up to 100 milliseconds across the cortex. Those have to be trained too in order to link up things that are happening in different parts of the cortex that are all related to the same experience you've had. And so this could be the mechanism. So here's the working hypothesis. Okay, I'm going to say, this is now very speculative, okay? So two tiers of cortical processing. The one that you're familiar with, fast sensory motor processing, is almost certainly carried by a rate code. That's what happens over that 1 second that I showed you earlier, right? That's, it's almost, at this point, it's in all the textbooks, so it must be right. Okay, but I'm saying that there's another tier on top of that carries the long-term working memory, which is induced by traveling waves. And that it triggers STDP, and this lasts for hours. And this is going to be induced by spike timing. Now, there's a huge argument that has gone on for decades and decades between colleagues of mine, respected colleagues, who say spike timing is irrelevant. It's only the number of spikes, it's the rate. And they're right about, I say, yes, you're right about this layer here, fast sensory, but that's what you studied for your entire life. But, with one electrode, you're never going to see a traveling wave. Sorry. And so now, and by the way, STDP has been around for a long time, and traveling waves have been around for a long time. But we did a review. It was published in, I think it was 2017. In any case, we collected over 100 papers, experimental papers, of traveling waves, and you saw the one from the hippocampus, but there are 100 other papers out there. These papers have been, even though people know about them, they've been ignored. And the reason is nobody has ever come up with a reasonable function for traveling waves. And so we basically say, well, that's somebody else's problem. But no, they may be there for a purpose, and here's a hypothesis which actually solves a very difficult problem that we otherwise don't have any way to explain. Now, if this is true, that means that we have a real global workspace. We have a way for connecting up all the different parts of the cortex, and this could be the basis of thinking. That's what this essay is about, thinking about thinking. And if you think about this for a moment, this is the Holy Grail, right? We've all wondered. I know so many of my neuroscience friends got interested because they were intrigued by this problem. How is it that I think, and how is it that I'm aware of things, and so forth? And, we haven't had good ideas. Global workspace probably comes the closest, but that's not a mechanism. There are no neural mechanisms there. It's just a way to say something has to happen that's on a global scale. Well, these are mechanisms that can be tested. Now, here's another very controversial topic. If you talk to experts in language, linguists, and people in cognitive science, what they'll say is large language models, they don't understand language, that they are not, they don't think, right? And I'll tell you, it's hard to believe that it doesn't understand something when it makes sense. But I can be absolutely certain that they can't think the way humans think. And here's the evidence. You have a dialogue, and it sounds like it's a human, right? I mean, it passes the Turing test. I've written a book about this, by the way. But what happens when you stop? It goes silent, blank. There is no self-generated activity, the kind that you have when you're sitting by yourself and thinking. So it doesn't have the ability to generate its own internal thoughts. Well, let's add traveling waves and fast weight changes, and see if we can get them to think. So this is, I want to take you now to, if I have time, how are we doing on time? Okay, I'm almost done. This is the 2017 paper, Attention is All You Need. It was a paper about the transformer. It came out of Google. Google ignored it for a decade, but eventually OpenAI used it and got it out into the real world. Now, there's a box here called multi-headed attention. It's a matrix, basically. And you can see the input comes into two. It actually bifurcates, one into this matrix and the other one into the stream, and they're combined, added and normed. Okay, well, it's a matrix. There's n inputs, it's n squared, so it's really a bottleneck because it takes a lot of memory. And self-attention is what's used to form association between words. Like if there's a this in the sentence, what word is that associated with? And it really is the temporal context that we were talking about earlier. So the question is, can you replace it with traveling waves? And of all things, it had already been done by an engineer, a group at Stanford, Christopher Ree. And what he replaced it with was a linear state space model. It's linear, and this same model is used by engineers, both signal processing and control theorists, to model complex systems and complex signals. And there's a matrix A. This is now going to be like the self-attention matrix. And it turns out that when you use a matrix with a particular form, that I'll show you in a moment, that the performance, as you can see here, on models that range from 100 million parameters up to a few billion parameters across a dozen different tasks. The one that's the model, including this is a ChatGPT 2 era. These are relatively small networks, but you can see the bolded ones have the best performance, and it's the Mamba. It's the one with the traveling waves in it. Wow. Well, traveling waves are good. Maybe they're good for the humans. Maybe they're good for large language models too. Now, what kind of matrix did they use? They used a matrix that had a form that mathematicians call tuplets, and that means that they have the same value along each diagonal. So here's the main diagonal. It's the same A0, A sub 0, A sub -1, -2. So there's only n parameters. And it turns out that you can train this much faster. It has many fewer parameters. And in inference time, it is much faster by order of magnitude. So that means that it saves a lot of money, saves a lot of energy, and the only thing, it still doesn't have self-generative capacity. And that's why we have to add fast wave changes to it. Now, one last thing. I showed you this earlier. And the question is, what's really going on in the basal ganglia? Okay. Well, I told you it's topographically organized, and so the thalamus to each of the parts of the cortex has linked to a particular part of the striatum in the basal ganglia, and there's feedback, through the thalamus back, and there's traveling waves in both the cortex and the basal ganglia. So remember that diagram I had in which I showed that in each module there is a self-attention term. It may well be that the self-attention is actually in the basal ganglia rather than in the cortex, or maybe in both. But in any case, it's interesting that we have something here. And by the way, it comes back and it's combined with the feed forward, right? Just the way it is in the transformer. So there's a lot more here. This is just the beginning, scratching the surface. Next steps. We want to manipulate the traveling wave selectively, and that can be done with drugs or transgenics so that we can selectively knock out the traveling waves without changing the EI balance or anything else. That's one of the goals. The mouse has 2.5 square centimeters of cortex. We want to scale up our model, the ring model, to a 1 square centimeter, which is getting closer to biological scale, and implement STDP and long-term working memory, and give it the same tests that we give to the kind of things that humans can do. And finally, there's going to be a lot of room here for a lot of other things that can be explored. We know that neuromodulators are very important for regulating memory and also for what happens during when you're remembering sequences. Training on sequences involves the VTA, ventral tegmental area, with reward prediction error. Prediction error. Here's another case, just like with transformer, prediction error takes you a long way. So these are the people I've worked with. Lyle Muller is the one who worked on the sleep spindles. Pat Churchman is a longtime collaborator. And here's Andy and Max, who helped with the machine learning. And here's the neuron paper I didn't tell you about. Thank you.
Perfect timing. We have a 15 minutes break now, but I'm sure Terry will be here in the break answering some questions. So again, a big applause to Terry Sejnowski for his Brain Prize lecture. Thank you.